Assalamu alaikum and welcome everyone to our molecular biology ASCP preparation series. This program is all about providing real life MCQs for your certification success. The MCQs we'll discuss today and in upcoming season have been carefully collected from several years of past exams to give you the best preparation possible. I'm Dr. Loai, and I'm really excited to share my knowledge and experiences with all of you through this series. I also encourage everyone taking the test to share your question with us so that everyone can benefit. All right. So today we are starting with the first part of molecular science and our focus will be on nucleic acid chemistry. So let's dive in together, but don't forget to subscribe to our channel to receive all the latest lecture. Are you ready? Let's get started. In this series of molecular biology ASCP preparation, I will cover various topics of molecular science such as nucleic acid chemistry, basic molecular theory, biochemical reagents, and human microbial genetics. These topics collectively make up 20 to 25% of the total exams. I will simulate the test as a real exam using multiple choice questions and provide explanation for the answers afterward. So let's dive into the details of molecular science part. Here is the fourth section of molecular science part, include nucleic acid chemistry, basic molecular theory, biochemical reagents, and genetics part. As I said, these topics collectively make up 20 to 25% of the total molecular biology ASCP exams. All right. So let's begin with the first topic in molecular science part, nucleic acid chemistry. So in the first part of nucleic acid chemistry, we'll explore MCQs on the sugar, nitrogenous basis, and chemical structure of DNA, where we will find the correct answer together and explain them in detail. Okay, so let's start from the first question here. What I will do, I will read the question, and I want you please to read the options carefully. Then I will give you 10 seconds before moving to the answer slides. But you can pause the video to verify your answer before proceeding to the next page. All right? So let's go and read the first question. The first question say, what is the main difference between the sugar in DNA and RNA? I will give you 10 seconds. Okay? 3, 2, 1. So the correct answer is B. DNA contain deoxyribose while RNA contain ribose. As we know, DNA contain deoxyribose, which likes oxygen atom at the 2' positions, where our RNA contain ribose, which has a hydroxyl group at the 2' position. Okay, so let's move to the next question. Okay, the question said, which carbon of the sugar in DNA is linked to the phosphate group? 10 seconds. The correct answer is 5' carbon. Actually, in DNA, the phosphate group is linked to the 5' carbon of the deoxyribose sugar. This linkage is the crucial for forming the sugar phosphate backbone of DNA strand. Okay? Alright, the next question is, which carbon atoms in the sugar molecules are connected by phosphodiester bonds in DNA and RNA? 10 seconds. Alright, let's move on. Alright, the correct answer is 3' and 5'. Alright? Actually, in DNA and RNA, the phosphodiester bonds connect the 3' carbon atom of one sugar molecules to the 5' carbon atom of next sugar molecule. Alright? These form the sugar phosphate backbone that is crucial for the structural integrity of nucleic acids. Alright, let's move on. Okay, the next question. What type of bond is formed when the hydroxyl group of one sugar molecule reacts with the hydroxyl group of another sugar molecule? The correct answer is glycosidic bond. Actually, when hydroxyl group of one sugar molecule reacts with the hydroxyl group of another sugar molecule, a glycosidic bond is formed. This reaction results in the release of a water molecule and the creation of an oxygen bridge between the two sugar molecules. This process is known as the dehydration synthesis or condensation reaction. All right, let's move on. What is the bond formed between the two deoxyribose pentose sugars connecting each other to form the backbone of DNA? Focus and focus and focus. Let's move to the result. The bond formed between two deoxyribose pentose sugars connecting each other to form the backbone of DNA is called a phosphodiester bond. This bond involves the phosphate group linking the 3' carbon atom of one sugar to the 5' carbon atom of next sugar in DNA strand as we see in the image. Alright, let's move on. Alright, the next question is the bond between a phosphate group and the ribose sugar group in RNA is called which of the following? 10 seconds. Let's go. Alright, let's move to the result. Alright, the bond between a phosphate group and the ribose sugar group in RNA is called a phosphodiester linkage. This important linkage forms the backbone of RNA molecule, connecting adjacent nucleotides through strong covalent bonds. Think of it as the glue holding the RNA structure together. It works by linking what? The 5' carbon of one ribose sugar to the 3' carbon of next ribose sugar. So this creating a continuous chain. This chain is crucial not only for the stability of RNA but also for its ability to perform various functions in the cell. All right? So without this structure feature, RNA would not be able to maintain its integrity or carry out its role effectively. All right? Let's move on. Okay, the next question. A nitrogen base is linked to sugar by a glycosidic bond at carbon number. Which carbon number? All right, the sugar is linked to nitrogenous base at the first carbon by glycosidic bond. So the correct answer is the carbon number 1. Okay, let's move on. Okay, for next question, which carbon of sugar in DNA is missing an oxygen atom compared to the RNA? 10 seconds. So the all right. In DNA, the sugar deoxyribose is missing an oxygen atom on the 2' carbon compared to the sugar ribose in RNA. This absence of an oxygen atom on the 2' carbon distinguishing deoxyribose from ribose and is crucial for the structural difference between DNA and RNA. So the correct answer is carbon number 2. All right, let's move on. Okay, let's move to the next question. In RNA, the sugar ribose has a hydroxyl group attached to which carbon? 1', 2', 3', or 5'? All right. All right, the correct answer is 2' carbon. Actually, in RNA, the sugar ribose has the hydroxyl group attached to the 2' carbon, as we see in the image. This hydroxyl group is the key difference between ribose in RNA and deoxyribose in DNA. So the correct answer is option B. All right, let's move on. The next question is, what type of bond length the sugar and phosphate groups in the backbone of DNA and RNA? As I said earlier, the bond that links the sugar and phosphate group in the backbone of DNA and RNA is called a phosphodiester bond. So this bond formed between the 3' hydroxyl group of one sugar molecules and the 5' phosphate group of adjacent sugar molecule, creating the repeating sugar phosphate backbone. All right. So let's see the question here. Which of the following statements is true about the sugar in RNA? 10 seconds. So the correct answer is B. It has a hydroxyl group at the 2' position. Actually, this is defining feature of the ribose sugar in RNA, distinguishing from the deoxyribose in DNA, which lacks the hydroxyl group at the 2' position, as I said earlier. All right? All right. Which of the following component is found in both DNA and RNA? I think this is a simple question. 10 seconds. Actually, the component found in both DNA and RNA is adenine. Actually, thymine found only in DNA, uracil found only in RNA, ribose found only in RNA. DNA has a deoxyribose instead, and adenine present in both DNA and RNA as one of the nitrogenous base. Okay? Okay, and the next question is, adenine and guanine are examples of what class of nitrogen base? So the correct answer is purines. Actually, adenine and guanine are both purines, which are characterized by what? Characterized by their double ring structure. So that make them larger than pyrimidines, which have a single ring structure. All right, let's move on. So the next question is, which of the following nitrogenous base is a pyrimidine? The correct answer is cytosine. Pyrimidines are single ring nitrogenous bases which include cytosine, thymine in DNA, and uracil in RNA. All right, so let's move on. Which of the following nitrogenous base is found only in DNA? 10 seconds. All right. All right, the correct answer is thymine. Thymine is found only in DNA and pairs with adenine. All right, where in RNA, thymine is replaced by uracil. Okay, let's move on. The next question is, in RNA, which nitrogenous base replaces thymine found in DNA? The correct answer is uracil. In RNA, uracil replaces thymine and pairs with adenine. Okay? So the correct answer is uracil. Let's move on. So the next question is which of the following is not a DNA base? Okay, the correct answer is uracil. Uracil is not a DNA base, it is RNA base. Okay, let's move on. The next question is which of the following is not an RNA base? 10 seconds. The correct answer is thymine. Thymine is not RNA base, it is the DNA base. As we see in the image here, RNA contains four bases: uracil, cytosine, adenine, and guanine, where thymine is specific to DNA. Alright, let's move on. The next question is what type of bond is formed between the nitrogenous bases of the two strands of DNA? Okay, the type of bond formed between the nitrogenous bases of two strands of DNA is a hydrogen bond. This hydrogen bond play a crucial role in maintaining the double helix structure of DNA. All right, let's move on. Okay, the next question. What is the name of bond that links the nitrogen base and the ribose sugar in a nucleotide? 10 seconds. Okay, the bond that links the nitrogen base and the ribose sugar in a nucleotide is called a glycosidic bond. This linkage occurs between the, focus please, 9' nitrogen of purine bases or the first prime nitrogen of pyrimidines and the 1' carbon of deoxyribose sugar group. Alright? As a result of this nitrogen carbon linkage, nucleotides are formed, which are essential component of DNA. Okay? Okay, the next question. At which position on the ribose ring is the base group attached to nitrogen base group? Okay. Actually, in both DNA and RNA, the nitrogenous base is attached to the first carbon of ribose or deoxyribose sugar. Okay? This attachment is crucial for forming the nucleotides that make up the nucleic acid chain. Alright, let's move on. Okay, the next question is in a DNA molecule, the glycosidic bond connects the first prime carbon of deoxyribose sugar to which nitrogen atom in purine bases? 1379. The correct answer is 9' nitrogen. Actually, in purine bases, the glycosidic bond connect the first prime carbon of the deoxyribose sugar to the 9' nitrogen of the base. All right, let's move on. In pyrimidines bases, the glycosidic bond connects the first prime carbon of the deoxyribose sugar to which nitrogen atom? 10 seconds. All right. Oh, the correct answer is A. In pyrimidine space, I mean the cytosine and thiamine, the glycosidic bond connects the first prime carbon of the deoxyribose sugar to the first prime nitrogen of the base. All right, so let's move to the okay for the next question. How many amino acids are used to build proteins? So 10 seconds. Let's go. 20, 30, 40, 50. The correct answer is A. There are 20 standard amino acids that are commonly found in proteins. These amino acids differ from each other in their side chains, which confer unique chemical properties to each amino acid. Alright? So these 20 amino acids are building blocks of protein and are essential for various biological functions. Alright? Let's move on. The next question. Which DNA conformation is characterized by the right-handed helix with the deep, narrow major groove and a wide, shallow minor groove? You have to focus on this question because a little bit confused. When we say right hand with the deep, narrow major groove and wide, shallow minor groove, which one of these DNA? B, Z, A, or C DNA? You have 10 seconds. All right, here we go. The correct answer is C, aDNA. aDNA actually is the right-handed helix with the deep, narrow major groove and a wide, shallow minor groove. Okay, so let's move to the next question. Which DNA conformation is most commonly found in a cell and has a wide major groove? You have to remember wide major groove and narrow minor groove. Okay, which one of them? A, B, Z, or C DNA? 10 seconds. All right, so the correct answer is B, bDNA. Actually, bDNA is the most common form of DNA found in cells. All right, it has a wide major groove and narrow minor groove. All right, so let's move to the next question. Which DNA conformation is the left? Is the left? Is the left-handed and often forms due to the stress or torsion during transcription? Which one of them? A, B, Z, or C DNA? You have 10 seconds. All right. The correct answer is Z DNA. Actually, Z DNA is left-handed helix. You have to remember, left-handed helix that can form due to the stress or torsion, such as during transcription. Okay, so let's move to the next question. What component of DNA is responsible for it is the negative charge of DNA? 10 seconds. All right. So the correct answer is phosphate group. DNA is composed of nucleotide, as I said earlier. Consists from what? Consists from the deoxyribose sugar, nitrogenous base, and phosphate group. Actually, this phosphate group is responsible for negative charge of DNA. Why? Because this phosphate group contains negative charged oxygen atom. All right. So when DNA in a aqua solution, this oxygen atom lost their protons. This lead to the resulting in negative charge. All right. What type of bond links the nucleotide in a single strand of DNA? 10 seconds. All right. So the correct answer is the phosphodiester bonds. All right. Let's move to the next question. How many base pair make up a codon? 1, 2, 3, 4. 10 seconds. Actually, a codon consists of the three nucleotide base in messenger RNA. So each nucleotide base represents one of four possible nitrogenous bases, even adenine, cytosine, guanine, or uracil in messenger RNA. Therefore, the correct answer is C, and a codon is composed of the three base pairs. All right. So let's move to the next question. Which of the following is not a component of a nucleotide? As we know, a nucleotide is composed of the three main components. Nitrogenous base, this can be adenine, thymine, cytosine, or guanine in a DNA, or adenine, uracil, cytosine, or guanine in RNA, plus pentose sugar. This is the 5-carbon sugar, which is the deoxyribose in DNA and ribose in RNA, and also include phosphate group. This group is attached to the sugar molecule and help form the backbone of the DNA or RNA strand. So the correct answer is anticodon, which is not a component of the nucleotides. All right. Next question. How many hydrogen bonds are formed between one A T base pair? 1, 2, 3, or 4? This is also the gift question when it comes in the exam. So the correct answer is 2. Actually, in DNA, adenine base pair with the thymine through the two hydrogen bonds. This specific pairing help maintain the stability and structure of DNA double helix. All right. All of the following are components of nucleic acid except one of them. Which one? 10 seconds. Actually, we should pay attention to the question that contains the word except. In our question, the correct answer is D, formamide. All right. Formamide is not a component of nucleic acids. So the nucleic acid, as we say, such as the DNA or RNA, are composed of phosphate group, sugar group, and the third component is the nitrogenous base. All right. So the formamide is not a component of nucleic acid. That's all for our lecture today, inshallah. See you in the next lecture. Assalamu alaikum. Please don't forget to like, comment, share, and subscribe to our channel to receive all the latest lecture. Bye.